Welcome to Chapter 13. In this chapter, we're going to discuss the microbe-human interactions. And in this particular picture, we actually see an eyelash, and the eyelash is right here. And then we see some bacteria on the surface of the skin, and then we have these little dudes. These actually are what we call follicular mites, and they are found primarily in the hair follicles of coarse hair, such as the eyelashes. And we actually want them there because these are part of our normal microbiome. So let's define some terms first. So what is an infection? An infection is a condition in which pathogenic microorganisms penetrate the host defenses, enter the tissues, and begin to multiply within those tissues. Pathologic state is the cumulative effects of infection damage. It is the disruption of the tissues and organs, and this results in disease. So what exactly is disease? Disease is any deviation from health. Factors that cause disease are certainly infections by microbes, but diet, genetics, and even aging. Infectious disease is actually disruption of the tissue or organs caused by microbes or by their product. Now, our normal or resident microbiota. This is a large and diverse collection of microbes living in and on the body. And we want these microbes present because they are going to keep other microbes that are pathogenic many times from invading. Also, this group of microbes is known as the resident or indigenous microbiota, or an old term was actually normal flora. This includes a variety of bacteria, fungi, protozoa, and even viruses. These organisms have had a profound effect on human biology. It is estimated that for every cell of your body, you have 10 microbiota present on the surface of the body or in the body. So, the relationship among the resident, transient, and disease-causing microbes and the host is that the resident microbiota oftentimes form a barrier for colonizing microbes that could cause disease. Now, some of these normal microbes will actually cause disease under the right condition, but for the most part, these are actually going to be protective in nature. So preliminary results of the Human Microbiome Project actually shows that human cells contain, again, somewhere between 22 to 25,000 protein encoding genes, while the microbes that are in and on us actually contain 8 million genes. So we have a lot of microbes in places that we used to think were actually sterile. All healthy people harbor potential dangerous pathogens, but typically these pathogens are in such low numbers that they are kept in check by the other normal good microbiota. So table 13.1 shows sites that are known to harbor normal microbiota, and certainly the skin and mucous membranes. And this is very important in actually first line defenses of the body. The respiratory tract, and even down deep in the lungs, the GI tract, the outer opening of the urethra, external genitalia, the vaginal canal, external ear canal, and even the eye. Additional sites, however, that we thought at one time were actually sterile, but now we know that they actually harbor at least some normal microbiota, include the lower respiratory systems down into the alveoli, the bladder and urine, breast milk, the amniotic fluid that surrounds the fetus and even the fetus itself, and sites in which DNA from microbiota has at least been detected, if not the microorganism itself, is actually the brain and the bloodstream. Now, benefits of normal microbiota include the influencing of the development of organs. It prevents the overgrowth of harmful microorganisms that try to invade. And this is through the process known as microbial antagonism. The general antagonistic effect of the good microbes they have a good effect against these intruder microorganisms. And typically this is going to be through the production of some sort of antimicrobial from the good guys against the bad guys. The microbes in the microbiome actually are in a steady, established relationship and therefore are unlikely to be displaced by any incoming microbes. Now, the gut microbiota. The makeup of your intestinal microbiota can influence many factors of your overall health. Differences in gut microbiome have been preliminarily associated with differences in risks for Crohn's disease, obesity, heart disease, asthma, autism, diabetes, rheumatoid arthritis, and even our mental health. So thoughts, moods, and the propensity for mental illness may actually have a link to the type of microbiota that you harbor in your gut. So, factors that weaken the host defenses and increase susceptibility to infection certainly age, the very young and the very old. The reason for this is the immune function is not actually working very well at that point. Genetic defects in immunity, either acquired or congenital. Surgery and organ transplants. Underlying disease such as cancer, liver, kidney disease, diabetes, chemotherapy or immunosuppressive drugs, mental and physical stress, or even other infections. Endogenous infections are caused by the microbiota already present in the body. We have E. coli in our gut. As long as E. coli stays in the intestinal tract, it causes no problems. But could it potentially get to other sites in the body? And yes, it can, and there it can cause problems. So this can occur when the normal microbiota is introduced to a site that was previously sterile or did not have that particular organism. So, an example, E. coli entering the bladder resulting in a urinary tract infection or UTI. Now, when exactly does the initial colonization begin of the newborn? Well, a growing number of doctors and scientists believe that actually this starts in utero, that fetuses may be seeded with normal microbiota from mom actually while they are developing in the uterus. However, and I will say, these microbes are important for the healthy full-term pregnancies and healthy newborns. But we know for sure that exposure occurs during birth with the baby becoming colonized as it passes through the vaginal canal. Now, one of the things that we have tried to do is with C-sections is actually cause, instead of just going in and taking out the baby, we actually cause the head to pop through the incision. And part of this is to help colonize, especially the respiratory and oral cavities, with normal microbiome found on the mother's skin. Continued colonization of the baby will be through their diet. Breast milk contains around 600 species of bacteria and sugars that babies actually cannot digest. The sugars are there to actually help feed the bacteria. So these sugars actually help establish the normal healthy gut bacteria. Breast milk may also be necessary for maintaining a healthy gut microbiome, at least within the first year. So, previously thought to be sterile was we actually thought the amniotic fluid and amniotic sac was sterile, but probably it actually has its own microbiota, and part of that may be actually passed to the growing embryo and fetus. Birth, certainly vaginal births, we're going to get a lot of microbiome from the vaginal canal into the nasal passages, oral passages, even the eyes. And that's why, again, C-section deliveries, we actually are trying to allow the head to pop through that incision that's made through the wall of the abdominal cavity in order to at least get some of those microbes from the skin. Certainly breast milk and even formula that has actually been uh, made to have some of these microbes in it is very good. And then who handles the baby? And of course, their environment. So don't bother to learn the chart, but I'm just going to go through a few things here on these sites that contain well-established normal microbiota and some representative samples. So on the skin, we have a number of bacteria, both gram-positive and gram-negative, again, along with some yeasts and even small protozoans and even small mites, such as the follicular mites. So we have staph, including Staphylococcus aureus, but Staphylococcus epidermidis is the primary staph we see on the surface of the skin. In the oral cavity, there's a whole slew of organisms that can be found in the oral cavity. In the intestinal tract, one of the big ones that we see is this guy right here, and that is lactobacillus. Lactobacillus is actually found on several areas of the body and are very protective. In the respiratory tract, again, a whole slew of organisms that we normally will see. The vaginal canal oftentimes will have lactobacillus as a predominant component. It will also, though, usually have some candida, and that's why a lot of times broad spectrum antibiotics will kill off the lactobacillus and cause the candida then to overgrow. Lactobacillus is also a predominant organism in the urinary tract. So, a condition in which pathogenic microorganisms penetrate host defenses, enter the tissues, and multiply is called what? The Human What Project collects genetic sequences of microbes from many different body sites. True or false, the lungs are sterile sites within the body? True or false, your gut microbiota can determine even your mood and mental health? And list and describe 3 factors that weaken host defenses and increase susceptibility to infection. And true or false, babies born by cesarean section are colonized by the same microbiota as babies born vaginally. Now, what is a pathogen? Well, a pathogen is anything whose relationship with its host is parasitic. We say that it's pathogenic, meaning it can cause infection and disease. The type and severity of infection depends upon both the pathogenicity of the pathogen and the condition of the host. So pathogenicity describes an organism's potential to cause infection or disease. So we could say something is extremely pathogenic, such as rabies, or we could say that the rhinovirus has a low pathogenicity. True pathogens are capable of causing disease in healthy individuals that have a normal immune defense system. Opportunistic pathogens cause disease when the host defenses are compromised in some way or when they become established in a part of the body that is not natural to them. Virulence is the relative severity of the disease caused by a particular microorganism. So again, rabies is an extremely pathogenic organism. It's also highly virulent. Virulent factors are any characteristic or structure of the microbe that can contribute to toxin production or introduction of an injurious host response. And something that goes along with virulence is the infectious dose. This is the minimum number of microbes required for an infection to proceed. So if I have a highly virulent organism, I will not have to have very many organisms in order to establish an infection. But if I have a low virulent organism, the infectious dose must be much greater. And this has actually been determined experimentally for many microbes. Microbes with greater virulence then need only a small infective dose. And in fact, rabies can be caused by a single rabies virus. So this picture just shows what can happen with the microbe and the host. And so there is a combination. So if the virulence of a microbe is low and the percentage of optimal infectious dose is relatively low, and there's no correct portal of entry for it, and the immune system of the host is working well, then probably the microbe will pass through completely unnoticed. However, if virulence and dosage go up and there is a portal and the immune system of the patient is actually good, the microbe may pass through unnoticed, or it could actually become established without actually causing disease. And so it could colonize but not necessarily cause infection. Now, if we have a medium virulence and a fairly low dosage and the correct portal of entry is not present, but we have an immune compromised patient, again, perhaps the microbe may pass through unnoticed, or we may become established without disease. But if virulence is high, infective dose is high, portal of entry is open, and the patient is immune compromised, then most oftentimes microbes will actually cause disease. Now, I want to mention here when we talk about infectious pathogens, we can actually study these, but we want to study them safely. And so the CDC has actually given us a biosafety level laboratories, and there are 4 levels of these biosafety labs. This is a system then of biosafety categories adopted by the CDC based on the general degree of pathogenicity and the relative danger in handling the pathogens in the laboratory setting. So, a biosafety level 1 lab. This is a standard laboratory open bench. There's no special facilities and no PPE is required, personal protective equipment. And typically here we're working with agents that have a low infection hazards. Most of the time their microbes are not generally considered pathogens and typically would not colonize the body of healthy individuals. A level 2 lab has the personal protective equipment. So laboratory coats, gloves, safety goggles, safety cabinets may be needed. Biohazard signs will be posted and access will be restricted. And this is when we start to work with agents that have a moderate potential to infect. So class 2 pathogens can cause disease in healthy people, but can be contained with proper facilities. So most pathogens that we deal with belong to class 2 and will be studied in a level 2 class laboratory. And this would include staph, E. coli, salmonella, etc. However, when we get to a class 3 microbe in a level 3 lab, now we have personal protective equipment. Restriction or access restriction will be in place, biohazard signs, but now we actually go one step further, and most experiments will be actually performed in safety cabinets or underneath ventilated hoods. Agents in the class 3 will be those that could cause severe or lethal disease, especially when inhaled. And that's why we're working with safety cabinets or collective hoods. And then class 4 level safety labs, this will have all of the minimums of the safety 3, plus you actually now will be in your own protected environment in a hazmat suit. So these are what's known as shower in, shower out facilities. When you go into a level 4 lab, you would actually go into an outer area first. You would shower, you would put on your hazmat suit, You would then go into airlock basically, where pressure is going to be changed, and you will either have your own oxygen on your suit, or as soon as you enter into the lab proper, you will hook to the oxygen system of the lab. Nothing will go in or out of this lab from a microbe standpoint that has not been incinerated. The labs are usually operating under a lower than atmospheric pressure, so if there should be a breach of the outer walls, air will flow in and not out. Level 4 organisms are those that are highly virulent microbes that pose extreme risk for most morbidity and mortality, especially when inhaled in droplet or aerosol forms. And so things like Ebola, the flaviviruses, some of the arenaviruses, all of these will be looked at in a level 4 laboratory. Now, the steps involved when a microbe causes disease in hosts, we can actually see this progress. First of all, as I mentioned in the slide previous, we have to find an open portal of entry. And that actually can sometimes be difficult. But I will say, most organisms actually enter the body either through the GI tract or the respiratory tract. They can, however, enter the body through any open orifice. Then they have to attach or adhere. They can do this by using fimbriae, the capsule, surface proteins, viral spikes on the virus capsid or envelope. They have to actually sit down on a surface within the body. Then, if they can survive the host defenses, so they have to figure out ways to avoid phagocytosis. They have to avoid death inside the phagocyte if they are phagocytized, and how to avoid the immune system response. And if they do this, they then become established and begin to reproduce, and this is when they then cause a disease. These organisms can cause disease by direct damage via certain enzymes or toxins they produce. They could produce damage by causing an excessive host response of the immune response, or they could actually cause epigenetic changes in the host chromosome. And this is certainly true of viruses. Now, eventually, usually most of these organisms will need to exit the host in order to infect another susceptible host. And portals of exit actually are usually some of the same ways that they get in. And so, again, respiratory tract, salivary glands, even sloughed off skin cells, feces, urine, blood, all of these will be exit points. So we'll look at each one of these in a little detail. So portals of entry, as I said, can be any open orifice to the outside, or it could be through a break in one of our physical barriers, the skin or mucous membranes. If the pathogen is exogenous, it means it originated from outside the body. If it's endogenous, it typically is one of the normal microbiomes that now gets introduced into either another area of the body, or this has been a previously silent infection. So this picture just shows some of these portals of entry. And certainly, again, broken skin, an insect bite, even the ear canal could be a potential portal of entry. So sites of entries in through the skin or mucous membranes could be due to nicks, abrasions, punctures, and sometimes these punctures are extremely tiny and even inapparent. The conjunctiva of the eye. But I will say this, intact skin is a very, very tough barrier that very few microbes can penetrate without having some sort of physical damage already done to the skin. The GI tract. We oftentimes will get our pathogens entering the body through the GI system because they're in the food or drink or other ingested substances. Oftentimes, these pathogens are adapted to survive digestive enzymes or abrupt pH changes, such as the acid of the stomach. However, very few actually are able to survive, especially the extreme acidic environment of the stomach. The respiratory tract is another major area of uh, entry, and this can actually be through the oral cavity itself or through the nasal cavity. Now, we have the oral and nasal cavity lined by continuous mucous membranes, and then that continues on down through both the GI and the respiratory tract. Even the auditory tubes that lead to the middle ear are lined by this mucous membrane. Two things. The mucous membranes provide a physical barrier because they're made of stratified squamous epithelium, but they also secrete the mucus that can help trap pathogenic organisms. So microbes often are transferred, though, from one site to another through sometimes this mucus as well. Urogenital portals of entry through the urinary system or the genital tract. Oftentimes we're looking here at the sexually transmitted infections or STIs. And these are pathogens that are transmitted in one way at least by sexual means or sexual contact. STIs actually account for about 4% of all infections worldwide. In the U.S., there's somewhere around 13 million new cases every year in the United States of sexually transmitted infections. Portals of or entry points through the skin or the mucosa of the penis, the external genitalia, the vaginal canal, even the cervix, and the urethra. So this table shows some of the common sexually transmitted infections. Now, trichomoniasis, caused by a protozoan parasite Trichomonas vaginalis, is actually probably the most common sexually transmitted infection seen in the United States. However, because oftentimes trichomoniasis is not diagnosed or reported, it is actually not the most commonly reported STI in the United States. That actually is chlamydiosis, with somewhere around 3 million estimated cases every year in the U.S. But we'll see papillomavirus, herpes simplex, and this is herpes simplex 2 that causes genital herpes, gonorrhea, hepatitis B, and I can put C in here, HIV, and syphilis. Now, the placenta is actually an exchange organ, but it does form what we call the maternal and placental barrier. The placenta is actually made up of contributions from the endometrial wall of the uterus and from the extra embryonic tissues formed from the original embryo. So it's formed by both maternal and fetal tissues. It separates the blood from the developing fetus from that of the maternal bloodstream. But it permits diffusion of dissolved nutrients from the mother's bloodstream into the fetal stream and gases into the fetal bloodstream, as well as the diffusion of wastes from the fetal stream into the maternal stream. A few microbes actually will cross this placental barrier and can be spread then by the umbilical vein into fetal tissues. Other infections can be transmitted perinatally as the child passes through the birth canal. We actually put a drop of silver nitrate in the eyes of newborn, and that is to actually hopefully cut down on some of the common infections that we might be seeing in the vaginal canal that are in low numbers but could be picked up as that bowling ball head gets squished through that straw of the vagina. So this just shows the placenta. And so again, the wall of the uterus, in fact, the epithelial wall of the uterus is the only endothelial or epithelial tissue that has blood vessels. The chorionic villi actually will form the fetal or embryonic blood vessels, and these will intersperse then with the maternal blood vessels. There is no actual direct connection between the maternal and fetal blood. In other words, there's no actual mixing of the blood. The only time that occurs is when the placenta actually separates from the wall of the uterus after birth. Some common infections that can be passed across the placental barrier can be seen in this mnemonic called TORCH. The T stands for toxoplasmosis, caused by the protozoan parasite Toxoplasma gondii. O is for other diseases, and this can include syphilis, Coxsackie virus causing hand, foot, and mouth disease, varicella zoster virus responsible for chickenpox and shingles, HIV, and even chlamydia. The R is for rubella, one of the forms of measles. C is for cytomegalovirus virus, which is one of the herpes viruses. And then the H is for other herpes viruses, primarily herpes simplex 2. Now, adhesion. I said that once the organism gets in through a portal, it needs to adhere somewhere in the body. So the adhesion is the process by which microbes gain a more stable foothold on host tissues. Adhesion is dependent upon the binding between specific molecules on both the host and the pathogen. A particular pathogen, therefore, is limited to only those cells or organisms to which it can bind. Once attached, a pathogen then can invade body compartments and begin to grow and multiply. Now, quorum sensing, like we see in biofilms, is also important oftentimes with this because there is sometimes a chemical communication between nearby bacteria that can be critical to the establishment of an infection. So here we see fimbriae and actually attaching to host cells, and this may then allow the bacteria to either move in between the cells or actually into the cells or to destroy the cells by the production of enzymes. The capsules can actually then stick to somewhere in the cell, or the viral spikes. And again, oftentimes the viral spikes of viruses actually are what attach to a host cell receptor that's for something else. The virus really hijacks it, and this then will cause endocytosis to occur and actually bring the virus into the cell. And this table just shows some of the general ways that some bacteria will attach to their host cells. So, for example, Neisseria gonorrhoeae uses fimbriae to attach to general epithelial cells. Mycoplasma, which causes atypical pneumonia or walking pneumonia, actually has specialized tips at the bacteria that fuse tightly to the epithelial cells of the lung. There's some more fimbriae, viral spikes with influenza. Even in poliovirus, the capsid proteins themselves actually cause adherence. So once we get in through a portal and actually adhere to a surface in the body, we then have to survive the host defenses. Phagocytes are specialized white blood cells that engulf and destroy pathogens by means of enzymatic and antimicrobial chemicals. Antiphagocytic factors are oftentimes virulence factors used and produced by the pathogens to avoid the phagocytes within the body. So they circumvent some part of the phagocytic process. Either they're not phagocyzed at all, such as those encapsulated microorganisms. The phagocyte cannot recognize the organism because it's surrounded by the capsule, or they figure out some way not to be destroyed once they are phagocytized within the phagocytic cell itself. Virulence factors are structures or products or capabilities that allow a pathogen to cause infection in the host. And adaptations of these virulence factors that a microbe uses to invade and establish itself in a host. Virulence factors determine the degree of tissue damage that will occur. And so several different things that organisms can produce. So I said the skin is actually a very tight barrier that will not allow many things through it. But some bacteria actually will produce enzymes that will break down the intracellular structure of epithelial cells and allow then a way for the bacteria to get into the underlying tissue. These are known as exoenzymes. Some bacteria will produce exoenzymes, but they'll produce other things called exotoxins. And these oftentimes break down the cells or, again, cellular components that will allow the bacteria either to get into the cell or destroy the cell completely, again, allowing a way for the organisms to enter the body. Or they can simply produce some sort of block, or they can produce something that will block phagocytic response. So again, the capsule, the phagocyte simply cannot recognize this as being, quote, foreign because the capsule literally works as an invisibility cloak. Therefore, the cell will never be phagocytized. Now, exoenzymes will be secreted by a number of different organisms, and they break down and inflict damage on the tissue cells. They can dissolve host defense barriers and promote the spread of microbes deeper into the tissues. And some examples of this would be mucinase that breaks down mucus, keratinase that actually breaks down keratin within epithelial cells, and hyaluronidase, which breaks down hyaluronic acid, which is one of the things that we find in the intracellular junctions of epithelial cells, and we can also find it in connective tissues. Bacterial toxins are specific chemical products of the organisms that are poisonous to something else. An exotoxin is secreted by the living bacterial cell and affects the infected tissue. And there are many types of these. Endotoxins are not actively secreted, but they are shed actually from the outer membrane of the bacterium. And so endotoxins are actually components of the outer phospholipid membrane of gram-negative bacteria. So exotoxins usually are toxic in fairly minute amounts. They are typically specific to a cell type. They typically are just small proteins. They're pretty unstable. They can be converted to toxoids. They can stimulate antitoxins. They usually are not pyrogenic, meaning they don't stimulate systemic fever. And they can be released by both gram-positive and gram-negatives. Endotoxins are typically toxic only in high doses, but that's okay. The gram-negatives have lots of these in the outer phospholipid envelope. They typically will cause systemic fever. They are the actual lipopolysaccharides of that outer membrane of the cell wall. They typically tend to be therefore very stable. They're not converted to toxoids. They won't stimulate antitoxins, and they typically are released by the bacterial cell via shedding or during lysis of the cell. And these endotoxins can actually make you very, very sick. Now, we need to talk a little bit about patterns of infection. So a localized infection. This means the microbe enters the body and remains confined to the site close to the portal of entry. So things like boils, like this little pretty thing right here, actually entered through a nick in the skin and actually is causing the reaction here within the area around the entry point. Fungal skin infections and even warts would be considered localized infections. Sorry about that. Went backwards there. So, for example, tuberculosis. Primary site is the lungs, but it can actually travel to other areas. streptococcal pharyngitis, and this is what's shown here in the picture. We actually will see a strep throat that then could potentially travel and cause something called scarlet fever, which causes scarring of the endothelial lining of the heart, the endocardium, along with the valves. And toxemia, and this is where infection remains localized, but the toxins produced by the infection are carried throughout the body. Mixed infections is basically biofilm. Several agents establish themselves simultaneously at the infection site. In synergistic infections, microbes actually cooperate in breaking down tissues to allow others that don't have that ability to actually get through the tissue. In other mixed infections, one microbe may create an environment that enables another microbe to invade, and this is actually truly the formation of biofilms. Polymicrobial diseases, then, that we oftentimes see are gas gangrene, wound infections, dental caries or cavities, and certainly human bite infections. Interestingly enough, cat mouths are actually contain the most pathogenic organisms to humans in bite wounds. Humans are second, and dogs are actually third. Now, primary infection is where the initial infection started, whether it be a focal or systemic infection. Secondary infection is where the primary infection moves to or is complicated by an infection by a different microbe. So, for example, we might have a urinary tract infection that causes a secondary vaginal infection. So patterns of infection, we can look at what we call acute infections. Typically, these come on very rapidly with signs and symptoms, but they resolve fairly quickly. And in fact, the conventional definition of an acute infection or inflammation is anything that lasts less than 2 weeks in duration versus chronic infections, which can come on rapidly, but more often than not, the onset is much more slow and persists over a longer period of time. And again, conventional definition of chronic is anything that lasts 6 weeks or longer. Well, what about those infections that may last 3 to 4 weeks in between the 2 to 6 weeks? Well, most of the time, these are simply acute infections that are highly pathogenic or are not being brought under control by treatment or the immune system very easily. But sometimes you'll hear the term peracute because it truly isn't acute by the definition, and it's not chronic by the true definition. Now, I mentioned signs and symptoms. Well, what is a sign? A sign is any objective evidence of disease that can be measured or noted or seen by an observer, whereas a symptom is subjective evidence of disease as sensed or felt by the patient. So I could say, I don't feel good. That's a symptom. It's like, well, where does it hurt? Everywhere. That's still a symptom. So let me ask the question, is pain a sign or symptom? Well, we'll talk about that here in just a moment. When you put the signs and symptoms of diseases together, we have what's known then as the syndrome. And oftentimes diseases have specific signs and symptoms that can actually be used to diagnose the disease. So let's look at signs. Can I measure fever? Sure, I can take your temperature. Can I really measure if you have chills? And really, what are chills? Well, I feel hot. Oh, wait, now I'm cold. Now I'm hot. I can't really measure that. Your temperature may not fluctuate at all, even though you're feeling cold or hot. I can measure septicemia. I can measure the amount of bacteria growing in your blood. But what about the pain, the achiness, the soreness, or it's like, man, it just doesn't feel right. And in fact, we sometimes call these, along with malaise, just what we call the ADR syndrome, just ain't doing right. We just don't feel like ourselves. Can I listen to your chest sounds, and can I determine as a practitioner whether I hear air sounds or fluid sounds in body cavities? Can I measure fatigue? I can't really measure it. I might be able to look at you and go, you look tired, but that really truly isn't a true sign. I can measure the amount of white blood cells. I can even measure the size of your lymph nodes, but I can't measure itching. I can't even measure a headache or nausea. Now, I can measure vomitus. I can tell you how much the patient vomited, what it looked like, all of that. I can't really measure nausea or even abdominal cramps. Okay. So if I can measure it, or I can truly see that it is a response and that response means this, then it's a sign. Otherwise, it's a symptom. So, what is inflammation? Well, inflammation is the earliest symptom of disease. Inflammation can occur without infection, but infection will always have inflammation. And in the next chapter, we're actually going to talk quite a bit about inflammation. Inflammation is going to be a rapid, nonspecific response in infected tissues or in damaged tissues. And inflammation will result in edema, which is an accumulation of fluids in the afflicted tissue. And sometimes, if this becomes chronic, can result in the formation of granulomas and or abscesses. And granulomas is the chronic form, abscesses are the acute form, where the body tries to wall off the collection of the inflammatory cells and wall off the infection from the surrounding non-infected tissue. Lymphadenitis is simply typically lymph nodes in the general area of the infection are going to actually increase in size as they recruit more immune cells, and so we'll actually have swollen lymph nodes. Signs of infection in the blood, I can actually see what's called a leukocytosis, and this simply means an increase in the white blood cells. Leuko is white, cyto, cell. We will see early on usually a neutrophilia, so our neutrophils will actually increase. Others will increase as well, but they may actually increase only in certain infections. Leukopenia is the exact opposite, where we have too few white blood cells. Septicemia is the general state in which microbes are present and multiplying and growing in the blood and are present in extremely large numbers. So if I have a septic patient, that means I have got microorganisms reproducing in the blood all over the place. Bacteremia simply means I usually have small numbers of bacteria present in the blood, but they are not yet undergoing multiplication. Viremia is the presence of the virus in the blood, but this would just be the virons. So whether or not they're actively multiplying in their host cells, I simply have the virus present in the blood. Now, some infections actually go unnoticed, and these are known as asymptomatic, subclinical, or inapparent infections. So the host is infected, and we are seeing some changes at the cellular level, but it's not enough to cause actual signs or symptoms, so it doesn't manifest as the disease. The patient experiences no signs or symptoms and therefore won't seek any medical attention. Most infections, however, will be attended by some sort of symptoms or signs. Now, the last stage of our microbe life is going to be exiting in order to find a new susceptible host. So, portals of exit are avenues of departure for pathogens to exit the host. And we can do this by the secretion of bodily fluids such as mucus, excretion through the kidneys or the digestive system, discharge, let's say vomiting, or even sloughed off epithelial tissues. So again, this picture just shows a lot of those portals of exit, and you'll notice they pretty much are going to be the same things that are the portals of entry. Even broken skin, where we have blood leaking out, can actually be a portal of exit. Now, respiratory and salivary portals, so the GI and respiratory system as exits, the escape media for most of these pathogens are going to be mucus. We can also see it in sputum, and so things that are in the lungs may actually come up in the sputum, nasal discharge, and other moist secretions, and this would include saliva and tears. Skin, the outermost layer of the epithelium, is always being shed off, and so some of the microorganisms may actually float away with the skin cells. It's estimated that between 80 to 90% of all household dust is composed of sloughed epithelial cells. And so a single person every day can shed several billion skin cells. And about every 40 minutes, about 1 million cells fall off of us wherever we are. Some intestinal pathogens, they may enter through the oral cavity and then cause irritation of the intestinal mucosa. That increases motility within the bowel, and we actually then will have a fecal exit. Oftentimes these organisms will cause diarrhea, and this will cause rapid exit of the pathogen. And sometimes that's actually a good thing. Sometimes the diarrhea is actually clearing the infection from the body. Oftentimes, our helminths will have their eggs or larvae released in the stool to be picked up orally by someone else. And the feces that contain pathogens create a huge public health problem in especially contaminated food and or water. I apologize, my dog just barked, so that was Bella. The urogenital tract, agents involved in sexually transmitted infections typically leave the host in vaginal discharge or semen. It can be a source of neonatal infections that infect the infant as it passes through the birth canal. So herpes simplex 2, chlamydia, candida albicans, and pathogens that affect the kidneys and or the rest of the urinary system can actually be discharged in the urine. Blood can be a portal of exit when it is removed or released through vascular puncture. So blood-feeding animals are oftentimes the most common transmitter of these blood-borne pathogens. And this would include things like mosquitoes, fleas, ticks, and blood-sucking flies. And so there's our nice little pretty picture of our tick. Now, sometimes microbes actually are persistent in the body. While they may not be causing disease signs or symptoms at the moment, they are still present in the body. And this is known as latency. This is a dormant state of an infectious agent. During this state, a microbe can periodically become active and produce recurrent disease. So a good example is herpes simplex 1, causing cold sores. Anyone that's had cold sores has herpes simplex 1. You will never get rid of herpes simplex 1. For the most part, it's in a latent stage. You're not seeing clinical signs or symptoms. And in fact, with herpes simplex 1, you cannot pass that virus on unless you are showing active lesions. Sequelae are the long-term or permanent damage to organs or tissues of the disease. So we could have something that might destroy liver cells, and those liver cells are not regenerated. Now, we can actually follow the course of disease by a graph. The incubation period is the time from the initial contact with the infectious agent to the appearance of the first symptoms. And I do say symptoms here. The prodromal period is when the earliest notable symptoms of infection appear, and that may just be, I just don't feel good today. The prodromal period may be extremely short, or it may be actually fairly long. But once the prodromal period is over, we then get the period of invasion. And this is after the microbe has entered, adhered, established itself, avoided the immune system, and the infectious agent begins to multiply at high levels and exhibit its greatest virulence within the body. And this is when you get sick. Hopefully, either through treatment and or your immune system, we will overcome the organism. And at that point, the organisms begin to die and we enter the convalescent stage. And this is when the patient responds to the infection and the signs and symptoms begin to decline. Convalescent stage may actually take a long period of time depending upon the disease process. So we can put it in a graph. The incubation period, the organism has entered and is adhering and beginning to get ready to multiply. The prodromal stage, when we see those first clinical signs and usually symptoms, and then towards the end of that prodromal stage, we usually are starting to see the first signs. Again, prodromal stage may be very, very short, so much so that sometimes we even miss it. Then we enter into the period of invasion. The micro numbers are going up as well as the intensity of symptoms, and really somewhere in here we're at the height of infection. Now, if treatment and or the immune system does not work, this can actually continue on up until we actually die. But hopefully that doesn't happen and the period of invasion kind of levels off and the organisms begin to drop and we'll enter into that period of convalescence. So we can actually overlay the growth curve of a microorganism onto this chart. So here we have that lag phase, the period of log growth, where the microbes are growing exponentially, the stationary phase, and then finally the death phase. Now, reservoirs are a permanent place for an infectious agent to reside. A source is distinct from a reservoir in that the source is usually not a permanent place. It can be used sometimes, but not always. A carrier is an individual who inconspicuously shelters a pathogen, either having a latent infection or an asymptomatic infection, and can spread it to others without knowing. I said with herpes simplex 1, it has only been shown that you can transmit the herpes simplex 1 virus when you have the active lesions of cold sores. So we used to think that herpes simplex 2, causing genital herpes, was the same, that you would have to have active lesions in order to pass it on to another person. However, we have found that some individuals may have had herpes simplex 2, had the active lesions, it entered a latent stage, and during the latent period actually was able to pass it on to a sexual partner. And so in this case, then the latent phase would actually be a carrier as well. So who or what can be living or reservoirs? Well, our living reservoirs are animals other than humans or arthropods. And so, yes, of the 2,000 some odd pathogenic diseases that are caused by microbes in humans, we share in common about 1,600 of those with animals. Certainly, another human could be a reservoir, and for especially those diseases that only occur in humans, humans are the reservoir of those diseases. So this could be either actively ill individuals that are passing on the organism or carrier states. Arthropods, and this includes both insects and arachnids, can be living reservoirs in the fact that they actually will be vectors so that they can carry carry the disease from one reservoir to another reservoir. So here's just showing some of these examples. An asymptomatic carrier, they're infected, showing no signs or symptoms of disease, and yet they're able to pass it on to other individuals. Incubating carriers, those that may be in that incubation stage or the early prodromal stage, when the microbes are just really getting established and just beginning to multiply, can actually also be a source of infection. Convalescent carriers. Do we still have some of the microorganisms present during convalescence? And yes, we certainly do. Or again, we could have these chronic carriers who shelter the infectious agents for long period of times after recovery and because of latency of the infectious agent. Or it could be the individuals that may have had an asymptomatic disease or just never respond to the disease yet are carrying the organism. And one of the best examples of a chronic carrier was Typhoid Mary. And it could also be that we have passive carriers. And oftentimes this is through mechanical transmission, meaning that we pick up something from one patient and take it to another via the surface of our skin or medical equipment. One of the biggest problems that was seen in transmission of infections from one patient to another several years ago was actually stethoscopes. Nobody ever thought about the fact that nobody was cleaning the bells of the stethoscopes in between patients. Now, animals as reservoirs and sources, again, some animals may also be vectors. In epidemiology, a living animal that transmits an infectious agent from one host to another is considered a vector. Now, the majority of vectors are arthropods, either insects or arachnids. If they are a biological vector, what this means is that they are actively participating in the life cycle of the pathogen. In other words, part of the pathogen's life cycle must occur in this biological vector before it gets passed on to another susceptible host. So they serve as a site in which the pathogen can multiply or complete its life cycle. Mechanical vectors are not necessary to the life cycle of the infectious agent. It is simply that these vectors mechanically carry the organism from one host to another. So, for example, mosquitoes can be biological vectors, bats, chickens, others can be biological vectors. In the case of rabies, any warm-blooded mammal can be a biological vector of rabies. But oftentimes, like flies that are not the blood-sucking biting flies, roaches, lice, all of these are mechanical vectors because they are actually not infected. They simply carry the organism on a body part and then touch you with that body part. Now, a zoonosis is an infection that's indigenous to animals but naturally is transmissible to humans. And most of the time, zoonotic diseases do transmit from the animal to the human. But there are some diseases that humans actually can transmit back to the animal. I used to never have to deal with MRSA in my domesticated dogs and cats. However, we're seeing a higher number now of MRSA cases in our pet animals because the MRSA was brought in by their human owners. So humans sometimes are the dead-end hosts of these zoonotic diseases and do not contribute to the natural persistence of the microbe. But that's not 100% true. So spread of disease of these zoonotic diseases is promoted by close association of humans with animals. And we probably have some of the closest association with animals than we've ever had in human history before. I mean, how many of us sleep with a dog and or cat or multiples thereof? How many of us are close to cattle or horses, sheep, goats, chickens, whatever? How many of us may have snakes as pets? So we actually have a much higher zoonotic factor than we've ever had before. So people in animal-oriented or outdoor professions are at greatest risk for zoonotic disease. And this is just a few of the diseases. So with viral diseases, rabies, and again, any warm-blooded mammal can carry rabies. Yellow fever, some of the other viral fevers, hantavirus is a kind of up and coming new emerging disease. It's actually shed in rodent feces and then picked up as aerosolized particles in the lungs. Influenza, most of our flus actually come or first developed in swine, birds, chickens, geese, and then moved to humans, mutated in the humans to then go from human to human. West Nile virus. Primarily West Nile virus, their host is wild birds. And this includes, interestingly enough, blue jays primarily. And the mosquitoes will bite the birds and then bite us and can transmit the West Nile virus. Bacteria, Rocky Mountain spotted fever, in fact, any of the rickettsial diseases, psittacosis, leptospirosis, anthrax, brucellosis, plague, salmonella, tularemia, the list goes on and on here. Ringworm. I've actually gotten ringworm as a fungal disease, it is fungus, from a domesticated cat that was showing no clinical signs. Cats can actually get the spore of the fungus on their fur, not show any clinical signs, and then pass that on to someone else. And I got it on my eyelid, which was really a unique place to have to try to treat. Toxoplasmosis, we'll talk about later on. Trypanosomomyosis, trichinosis, and tapeworms. Now, non-living reservoirs, microbes have actually adapted to nearly every habitat in the biosphere. So they're found in soil, water, air, every place. Most are going to be saprophytic and cause very little harm to us. So even though we're completely surrounded by these continuously, they're not going to be a problem. There's only a very few of them that are true pathogens, and some are opportunistic. So soil, water, air, the built environment, so our houses, our buildings that we work and live in. Now, communicable diseases. These are diseases that occur when an infected host can transmit the infectious agent to another host and establish infection in that new susceptible host. Contagious disease is where we have an agent that is highly communicable, especially through direct contact. So these two terms actually get used kind of interchangeably. Is a contagious disease communicable? Yes. Is a communicable disease contagious? Well, that actually kind of depends a little bit. Here's how I kind of think of it. Contagious diseases are communicable. However, communicable diseases of certain interest are also reportable diseases. Non-communicable diseases such as cancer, of course, do not arise through transmission from host to host. Now, how does transmission in communicable diseases occur? Horizontal transmission is the disease is spread through a population from one infected individual to another susceptible host. However, vertical transmission can occur, and this is where transmission from the parent to the offspring via the ovum, the sperm, placenta, or the milk occurs. Now, we can have direct contact or direct transmission, and this would include physical touching, kissing, certainly sexual activity, or it could be in close contact when droplet contact is actually occurring, or parental transmission via intentional or unintentional injection. Indirect transmission is where the infectious agent must pass from an infected host to an intermediate conveyor, so a vehicle of some sort, and from there to another host. So a fomite is any inanimate object that harbors and transmits the pathogens, and this can be something like doorknobs, telephone receivers, the elevator button, the button on the vending machines, Kleenexes. Whereas vehicles are usually natural non-living materials such as the air, water, soil, or food. Now, there is a specialized indirect transmission, and this is called the fecal-oral route. And I like fecal-oral better than the oral-fecal. The reason for this is the pathogen is passed fecally from the infected patient and will then be found in the water, soil, or food, and then it's picked up orally from that vehicle. And then we have vector transmission, either biological vectors or mechanical vectors. So again, vehicles are any inanimate material, non-living material commonly used by humans that could transmit infections: soil, water, air. Fomites are inanimate objects that harbor the organism and can transmit it, but usually when the person comes in contact with that fomite. So it's not a continuous source of infection. I can clean the doorknob. I can clean the elevator button. The fecal oral route, again, shed in the feces picked up orally. And there's several of these. Hepatitis A. Of all the hepatitis viruses, this is the fecal oral transmitted hepatitis. Amoebic dysentery. The amoeba is in fecally contaminated food and or water. Shigellosis. Typhoid fever. And there are several, and actually many others. So water and soil can harbor microbes that can sicken humans. They can also become temporarily contaminated with pathogens from humans. Now, I can actually clean soil. I could heat it, for example, to actually sterilize it. I can sterilize water. So they could be temporarily sterilized, but they're going to pick up something pretty quick. Can we sterilize the air? Well, no. Indoor air can serve as a support media for the suspension and dispersal of respiratory pathogens, fungal spores, viruses, everything, be it nuclear or droplet modalities. Droplet nuclei are actually dried microscopic residues created when microscopic pellets of mucus and saliva are ejected from the mouth in coughing and sneezing. Aerosols are actually suspensions of fine dust or moisture particles in the air that contain living pathogens. So, is there a huge difference between droplet nuclei and aerosols? Not really. It's just basically that one is dry and one's moist. And I love this picture. Now, healthcare associated infections. So these actually were used to be called nosocomial infections because these are infectious diseases that are acquired or developed during the stay in a medical care facility, so a hospital or just even being in a clinic setting for a period of time. Rates of HIAs can range anywhere from about 0.1 to 20% of all admitted patients. And I'm going to say, if a healthcare facility has a 20% rate of healthcare associated infections, they have a real problem. Healthcare associated infections, if you are admitted to a hospital, you have a 5% overall chance of contracting a healthcare associated infection, something that you get from the hospital, not something you came in with. Your length of stay in the hospital increases by almost 18 days if you get one of these healthcare associated infections. 1.7 million people per year get HAIs while they're in the hospital. And these can cost systems somewhere around $35 billion a year and actually will cost over a patient over $1,000 per admission. It is 9.4% of the total inpatient costs altogether. And HAIs will actually kill more people each year than breast cancer and prostate cancer combined. So are these a problem? They absolutely are. Our healthcare facilities are where the sick people come, and that's where we have huge populations of these infectious agents. So we want to practice medical asepsis, and that is to limit the spread of infectious agents from person to person. Again, Ignaz Semmelweis, that radical idea, wash your hands between patients. Surgical asepsis, ensuring all surgical procedures are conducted under the most sterile conditions that we can. Most facilities have at least one, if not more, infectious control officers, and these individuals implement proper practices and procedures throughout the healthcare facility. They are charged with tracking potential outbreaks, identifying breaches in both medical and surgical asepsis, and training other healthcare workers in aseptic techniques. And really one of our goals in the laboratory of this class is to teach you all universal protocols that are going to help you in the medical and surgical aseptic techniques. Now, we can even still use Koch's postulates to be or to study infections and disease in determining what's causing the problem, what's known as the etiological agent. And again, a little review, Robert Koch, remember, developed the standard for determining causation of a disease that really has stood the test of time throughout the scientific scrutiny. And he actually worked with anthrax and determined that the Bacillus anthracis was the etiological agent of cutaneous anthrax. And this is how he developed his four postulates. So again, remember we have to isolate from an infected patient and get a pure culture. We then have to inoculate that pure culture into a susceptible host and then re-isolate from that experimental animal and compare it to what we actually isolated from the original host and see if they are identical. So true or false, if a virus causing a respiratory disease is ingested, it will not cause disease. The minimum number of microbes required to cause disease is known as what? Bacterial blank are toxic to the host in minute amounts. And what is objective evidence of disease and what is subjective evidence of disease? During the period of what a pathogen exhibits its greatest virulence. And true or false, the etiology of all infectious agents can be determined by Koch's postulates. Now, epidemiology is the study of disease in populations. And have we not heard enough about the epidemiology of coronavirus lately? But epidemiology looks at the effects of diseases on community. It involves the study of the frequency and distribution of the disease and the distribution of disease and other health-related factors in defined population. Reportable diseases or notifiable diseases are those that by law are reported to some sort of healthcare authority. And so reportable or notifiable diseases here in Greene County are first reported to the Greene County Health Department, which in turn will share that information with the Missouri State Health Department as well as CDC. Now, there are some diseases that are only reportable to a county or state authority, but most of our reportable diseases are also going to be reported to the CDC. Other diseases are reported on a voluntary basis, and so these are actually not by law required to be reported. The Centers for Disease Control and Prevention, the CDC, located in Atlanta, Georgia, are responsible for keeping tracks of infectious agents in the United States. They are part of the U.S. Public Health Service, and they publish a morbidity and mortality weekly report of the notifiable diseases. And this is a weekly notice of diseases that are reportable in that year or over time. The CDC will share its statistic on disease with the World Health Organization for worldwide tabulation and control. And then there are some other reporting agencies as well that will also be involved with CDC and WHO. Prevalence of disease is the total number of cases at a given time in a population. So today I could say we have 1 million cases of H1N1 influenza in the United States. That's how many cases we have today. That includes cases that occurred before today as well as new ones that were reported today. Incidence of disease measures the number of new cases over a certain period of time. So I've been following Johns Hopkins University's COVID maps, and they report incidence rates. What incidence rates are the number of new cases every day over a period of time within a certain population. So I can look at the incident rate of coronavirus in Greene County. Incidence is also known as the case or morbidity rate, those that have the disease. Now, mortality, however, is how many die of the disease. So this measures the number of deaths in a population to a certain disease over a given period of time. So we can actually report epidemiological statistics in a variety of ways. We can look again at hepatitis incidence, so number of new cases over periods of years. And we didn't start actually tracking hepatitis C until actually about 1990. We can look at the percentage of people in the United States who received influenza vaccine in 2 recent years. So the green is 2010-11 flu season, the purple is 11-12 season. Or we could actually just simply see where are cases of a disease seen, and this is actually showing the prevalence of malaria in the world. And so we see that primarily this is an equatorial disease, and the reason for that is where the vector is located. Point source epidemics. Point source epidemics is one in which the infectious agents came from a single source. And so, for example, this is a point source epidemic traced to crab cakes that were served at a fundraiser in Maryland. So the fundraiser was from 3 to 6 p.m. on 8/22 in 2003. And we saw that there was uh, 2 or 3 cases that occurred on that day. And then we saw a few more cases occurring the next day and the next. Where were all of these people getting the onset of the disease, though? It all traced back to this one event that occurred. Common source epidemics result from a common exposure to a single source of infection that can occur over time. And this actually shows the outbreak of Legionnaire's disease in 1976 in Philadelphia at a Legionnaire's convention. And what they traced the Legionnaire's bacteria to were the water cooling towers in the or on top of the hotel. And so as they traced these cases, what they found is most of the cases occurred in the conventioneers. They were staying in that hotel. Legionnaire's disease is an airborne disease, and so it was actually being blown through the air conditioning system. But what they also found was that there were a few cases of non-conventioneers. And what they actually found was that those that did not actually attend the convention, though did have or were in close proximity to the hotel. They weren't in the hotel, but they walked by the hotel, or they were within a short block area of the hotel. And so what they thought was that water evaporating from the cooling towers on top of the hotel actually evaporated and then floated down into the surrounding environment through the air. A propagated epidemic results from an infectious agent that is communicable from person to person and is sustained over time within a population. So, right now, our coronavirus is a propagated epidemic. This actually shows the other coronavirus that was in 2003, and that is SARS virus. And so what we found was this was going from person to person over the period of time within the population. And then, of course, a pandemic is the spread of an epidemic across all populated countries. So, SARS was actually held within a few countries in 2003-2004. COVID-19 is a pandemic because we have found cases of it on every populated continent except Antarctica around the world. And so it is a pandemic. Oftentimes, we try to find the first patient found in an epidemiological outbreak. And this is sometimes referred to as the index case or patient zero. Endemic diseases are infectious diseases that exhibit a relatively steady frequency over a long period of time in a particular geographic locale. So, I will tell you, influenza is an endemic disease in the United States. It's always present. It tends to have lows and highs of number of cases, but it is always present within the population. Sporadic disease is where we see occasional cases of a disease reported at irregular intervals in just random locations, sometimes completely separated around the world. So we might see 2 or 3 cases in Alaska, and a few months later, 4 or 5 cases might pop up in Australia of the same case, with no linkage between the cases at all. So this is actually Lyme disease, and this was in 2010, and this is old data. I will tell you this right now. But the endemic regions for Lyme disease were up here in the upper Midwest and in the Northeast. And then we did actually see sporadic occurrence out here in the western United States. Well, the latest map that I've actually seen, that I couldn't find one to put in this presentation, this has actually pretty much spread, and really we actually now have endemic areas throughout the United States except around in the Rocky Mountain region of the U.S. And then this is actually the percentage of U.S. deaths caused by influenza and pneumonia over a 5-year period from 2005 to 2010. And we can see that there is an epidemic threshold, that we have these seasonal ups and downs, but yet overall, the disease is present at all times. And so again, this would be an endemic disease. Now, epidemiology looks at the global picture. We see increases in infectious disease incidence around the world. We have emerging diseases in certain parts of the world that are being spread to other parts. We have things like HIV that now are considered really endemic throughout the world, but when to begin with, actually spread from centers within Africa to around the world. SARS and COVID-19. Novel strains of influenza. About every 3 to 4 years, we get a new strain of influenza virus, primarily out of the Asian region of the world. Re-emerging diseases. These are diseases that we may have thought were kind of under control, but now we're seeing an increase for some reason. So things like dengue fever, tuberculosis, yellow fever, hand, foot, and mouth disease, whooping cough. And this is just a picture I found from 2017 of the global examples of emerging and re-emerging infectious diseases from around the world. And you can take a look at these, and there's a whole bunch of them. And we are so concerned about one particular disease right now, but I'm going to tell you, there's a lot more out there that will kill you a lot faster that we need to be very concerned about. One of the things we also need to be concerned about is bioterrorism, and this is the intentional or threatened use of microorganisms or their toxins from living organisms to cause death or disease in humans, livestock, or plants. Stakes are much higher with the advancement of biotechnology, genetic engineering, and genetic recombination. Genetic alteration of bioterrorism agents could actually cause devastating scenarios. For example, the Bacillus anthrax that was actually used in 2001 was an actually genetically modified anthrax that came out of a level 4 lab up in Ames, Iowa. How it got out, we still are not sure, because nothing should come out of that biohazard lab 4 unless it's been incinerated. Well, I can tell you, somebody walked it out, and we still don't know who that somebody is. Fortunately, this particular Bacillus anthracis had not been genetically modified so much that it caused extreme disease, but it still wound up killing a number of patients. And agroterrorism is actually kind of a subset of the bioterrorism, because think about this. Who controls the food controls the world. And so agroterrorism is the use of microorganisms to decimate the agricultural industry, both plants and animals. If I control your food source, I control you. So targets the food supply to exert damage within the food supply. And so far, knock on wood, no documented cases of agroterrorism have occurred, but many government agencies are conducting surveillance, and I guarantee you the U.S. is, and developing policies to prohibit this scenario. So, the number of new cases of disease over certain time periods is called what? The blank rate is the number of persons afflicted with an infectious disease. Air and disease is seen at a steady frequency over a long period of time in a particular location. And what is it called when it is an epidemic occurring over all of the inhabited continents? And blank diseases are caused by newly identified microbes. And so on that really happy note, in the next chapter we're going to start looking at our immune system. Thanks.